Let's start. My name is Robert Grass. I'm the host of this webinar, and today's webinar, which is kind of hosted by the DigNA DNA Data Storage Portfolio of the European Union, we are focusing on DNA synthesis. So Martin Jost and Mark Somoza will, in two parts, give you an introduction into the aspects of chemical DNA synthesis. Martin Jost, he's a co-founder and chief scientific officer at Kilobaser, a company that sells devices that make DNA synthesis. And Mark Somoza, he's associate professor of chemistry at the University of Vienna, and he has worked for the past 15 years on DNA synthesis on arrays. There's an interesting, let's say, side note. All three of us have a connection to Austria, where Martin and Mark both work in Austria, and I originate from Austria. And with that small side note, I'll allow Martin to start. I'll just, before I nearly forgot, this webinar is recorded and will be published on YouTube. If you have any questions to that, please reach out to me. And if you have any questions, please wait to the end, unless the speakers have other would allow that within their talk, and please ask questions via the question section, the Q&A chat, and then we'll collect the questions at the end. With that, Martin, may I ask you to start, please?
Yeah, thank you for the introduction, Robert. So let me start by giving you some more context on myself. As a kid, I enjoyed exploring software and got some early job experience as an autodidact programmer. But after finishing the technical high school, I realized I didn't want to work on other people's projects for my life. At the same time, I wanted to know how life really works on a molecular level. So I started studying molecular biology and some computer science in Graz in Austria. After some time, me and a few friends wanted more hands-on time in the lab, so we could work on our own projects. We founded Olga, a community lab for students and the general public, and a bit later we even obtained the license to work with genetically modified organisms there. After a few months, we stumbled upon a paper describing a microfluidic DNA synthesis chip. And by chance, we got access to a few €1,000 of venture capital, so me and my co-founders dropped out of university in 2014 to focus on Kilobaser. Against many odds, and with the help of early believers and investors, we managed to develop the basic idea to a product and bring it to market. And I believe we even introduced a few nice things to the field. In 2022, we joined the Dynamic Consortium in a journey to integrate decentral DNA synthesis capabilities into the autonomous robotic platform we're developing there. Since most of you probably have some interest in DNA data storage, you're probably not completely alien to the idea that DNA is a lot more than just a biopolymer. And this is also the thing that really excites me. It's actually one of the few ways we have to speak to the molecular world in its own language. So from my perspective as a computer nerd, DNA ticks all the boxes. It's of course the programming language of life, and everything is data. So in theory, we can use it to reprogram cells and make everything we want, or even start from scratch and design new forms of life. So yeah, by now I learned that things are in practice a little bit more tricky, but nevertheless, there are hundreds of examples where DNA is used exactly for this. A very prominent one are the COVID vaccines that really only provide the code or recipe and utilize the body's molecular machinery to make the actual antigen. DNA can be selectively copied and extracted, put together and inserted in other organisms. The classic example being human insulin made in microorganisms in bioreactors. Also, synthetic DNA is of course being used to guide the molecular scissors of CRISPR and similar techniques. Probably one of the most direct ways of interaction with the biological world on a molecular level. This was my personal reason to join the field, and I'm still quite excited of what we will be able to do with this power. And if you're interested in such visionary perspectives, I can warmly recommend the book The Genesis Machine, written by one of my first mentors, Andrew Hassel. So when we think of DNA, we all have a certain image in mind. DNA can be seen as a fine string with the naked eye. Under an electron microscope, the famous double helix becomes visible. In its natural form, it's a huge chain of bases, folded in the most amazing ways inside a tiny cell. But in the context of actually writing DNA, we think of it in a very different, quite abstract way. We don't care anymore about the well-known double helix, and not even the complementary bonding of A and G, C and D is overly interesting. Synthetic DNA is made as a single strand, a single chain of bases. We want to build it artificially, base by base. In order to do that, we have to solve some fundamental questions. First, how do we control the sequence? Chemistry works in liquids. If you put a molecule and potential reaction partners in a liquid, and the molecule has reactive sites, it will not react once, but instead form a chain or polymer as long as there are reaction partners in the same liquid. This is obviously not what we want. In chemistry, this is solved by protecting the reactive sites with protective groups. In the next step, some protective groups are removed by exposing the product to a deblocking agent with specific properties. Only then the reactive site becomes reactive again. Using another deblocking agent, we'll remove another protective group. So we can precisely control where the next reaction will happen. Specifically for DNA synthesis, we are talking about a backbone of circular sugar molecules, connected with phosphate groups shown here as green blocks. The well-known bases adenine, guanine, cytosine, thymine, short HECT, are attached at the other end of the sugar. So the principle is simple. D-block chain, couple next base. The next base then becomes the end of the chain. This is repeated in a cyclic fashion. D-block, couple, and so on. Now remember, all this takes place in solution. This means all chains float around freely in the liquid, while another liquid is being added to the mix. The issue is, for each step, the emergent chain or product has to be taken out of its solution, filtered, purified, and then submerged in the next solution for the next reaction step. If you're going to make a long chain base by base, this becomes very tedious. A very clever solution to this is tying one end of the chain to a surface, while flushing all the reagents over it. This is called solid phase synthesis, if you were asking yourself what this webinar's title was about. So with this, we can conveniently flush reagent over a surface, and the nucleotide chain grows without being flushed away. An actual solid phase looks more like this than a flat surface. It's a very porous powder filled into columns like this. But there are exceptions, as we'll see later today. Another thing to keep in mind, we are now talking about DNA in very abstract terms here. Like there's only one single chain on a single anchor. In reality, there are trillions of chains with exactly the same sequence on the same surface. DNA synthesis happens in the lab, usually in pico or nanomolar scale. This means about 10 to the power of 14, or roughly 100 trillion molecules are being made at the same time. So we have all ingredients now, and we can repeat this in a cyclic fashion over and over until we have a DNA chain with the sequence and length we desire. Problem solved. Maybe let's now go one step back and take a quick look at the history. In 1955, Michelson and Todd reported the first directed chemical synthesis of a D-nucleotide. These were two thymidine residues joined by a phosphate link. This was purely liquid phase chemistry, painstaking and low yield, but it proved that chemical synthesis of nucleic acid fragments was possible. In the 1960s, Hargoba and Corana developed the phosphodiester approach, which allowed the systematic assembly of defined oligonucleotide sequences. And these synthetic oligonucleotides were instrumental in cracking the genetic code, earning Corana the 1968 Nobel Prize. Corana's group also developed most of the protective groups still used in modern synthesis, such as the DMD group. This is bright red and can be used to monitor the progress of a synthesis in real time. Also, the groups protecting the nucleobases shown on the right side are still in common use. In the 1970s, Caruthers group achieved the first total synthesis of an artificial gene. This was a monumental accomplishment, but incredibly laborious one. Go back a few years. In 1965, Robert Letzinger introduced two transformative ideas. First, Letzinger introduced the phosphate tree ester method, and later the more reactive phosphate tree ester approach, which dramatically improved the coupling efficiency. But most importantly, he pioneered the solid-phase oligonucleotide synthesis, borrowing from Robert Merrifield's solid-phase peptide synthesis concept. Merrifield received the Nobel Prize for this in 1984. In 1981, Marvin Caruthers published the phosphoramidite method. He worked at this time at the University of Colorado and made what appears in hindsight to be a modest chemical modification of Letzinger's phosphate tree ester approach. He just swapped one leaving group for another. But this subtle change was pivotal. The resulting phosphoramidite building blocks were stable enough to be manufactured, stored as solids and shipped, and then activated on demand by simply adding a weak acid. This made commercial scale production and distribution of DNA synthesis reagents viable. Again, in the early 1980s, the collaboration between Caruthers and Leroy Hood at the Caltech led to the founding of Applied Biosystems, which commercialized the first phosphoramidite-based DNA synthesizer. Meanwhile, Serge developed the first commercially successful instrument, the Synthesis Automation Machine 1. Suddenly, labs all around the world had routine access to custom oligonucleotides. And the timing was also perfect. The invention of PCR in the 1980s created explosive demand for short DNA primers, driving the industry into a golden age. More recently, enzymatic DNA synthesis has attracted significant attention. The core idea is to use a template-independent polymerase, TDD, to add nucleotides one at a time in mild conditions in water. The principle was again proposed in 1962. That's of the same age as the phosphoramidite approach. It also follows the same principle. Nucleotides are blocked at the 3' end, and after each addition, the blocking group prevents further extension until it is removed. The appeal of it is clear in theory. Aqueous at room temperature chemistry, no hazardous organic solvents, and potentially the chain can be made longer than with phosphoramidite chemistry. However, the first commercially available synthesizer based on this technology was only released by a French company, DNA Script, in 2022. And it's very interesting that both approaches share a fundamental challenge. In cyclic chain extension reactions, the error or failure rate multiplies with each added base. So regardless of chemistry, it's extremely important to bring the yield of a single cycle to extremely high values of about over 99%. Otherwise, the approach does not lead to practical results. I will come back to that later. But first, let's have a closer look at the phosphoramidite cycle. Remember the fundamental concept. First deblocking, then coupling of the next base, which contains the next protected group for chain extension. In the phosphoramidite cycle, we have 5 steps: deblocking, activation, coupling, oxidation, and capping. A linker is attached to a solid surface. Either a linker or the previous base are protected with a DMD group. This is being removed by flushing with a weak acid. The activation step removes the protective group, blocking the phosphate group of the next amidite, shown here in red. The activation solution is mixed with the desired phosphoramidite, then flushed through the column. Now the activated amidite connects with the deblocked chain at the 5' end, forming a phosphate triester group. Oxidation of this phosphate triester group to a phosphate triester group strengthens the bond between the chain and the newly added nucleotide. Otherwise, it would be cleaved under the acidic conditions of the following deblocking step. And now here's an interesting part. The cycle actually contains a step to block unreacted chains from further participation in the next cycle. The capping step puts an acetyl cap on any unreacted but already unblocked chains. This is a very efficient reaction. and I'll come back to that later also in a minute. Finally, the solid support is exposed to basic conditions, often ammonia, to release the chain from its support. The product is then treated for an extended period of time to remove the remaining protective groups from the backbone and the nucleobases. This is typically done on a separate machine and is not part of the automatic synthesis cycle. Now let's come back to a few core concepts. Synthesis always happens in 100 trillion instances at the same time, if you remember. Some of this reaction always fail. Even with 99% synthesis fidelity, 1 trillion chains fail to extend with each cycle. This means no base is being attached. If the process continues, the next base is being attached at the unreacted site, leaving out the intended base. This introduces a deletion, which is in the final chain a mutation of the actual sequence we wanted. After some cycles, many kinds of different sequences will appear. The intended one is still dominant, but the number of mutations increases with each cycle. In the end, we have a mixture of various sequences in our final product. To make things worse, if only a single base is missing, the length is similar to the intended product. This makes separation and purification of the product much harder. If capping is introduced, the failed chains are not being extended, as you can see here. With capping, we still have truncated sequences in the final mixture, but for most applications, this is preferable over a wild mix of different sequences with deletions at several points. The multiplicative effect of stepwise yield can be nicely shown in this tabular form. In the left column, for chemical process already very good value of 98.5% leads to already 25% product missing after only 20 bases. After 50 bases, only half of the product is left. It does not make sense to go much beyond that length with such a yield. But even with an extremely high stepwise yield of 99.5%, 200 bases is quite the limit for any meaningful synthesis result. With phosphoramidite chemistry, 200 bases are typically only reached under optimal conditions by service providers, specifically optimizing for this use case. If one needs longer sequences or even plasmids with a few thousands of bases, this is done by making an oligopool using parallel synthesis of different sequences and then assembling them with enzymatic processes. So in reality, we never get pure product right out of any synthesizer. There are different methods like desorting, HPLC, gel electrophoresis, and so on to remove the garbage, but I won't cover them in detail here. The possible applications for this generated DNA have different sensitivity to impurities. For example, PCR is quite tolerant and even works with unpurified crude product. In DNA assembly for longer sequences, on the other hand, you want to have as pure product as possible, because errors will propagate even more. This means, depending on the application, there are quite a few laborious steps needed before you can actually use the DNA. Specifically for data storage, there are ways to circumvent this hard requirement or even exploit the properties of error characteristics. This is the expertise and goal of actually a few working groups within the DigNA portfolio. This is also an excellent example of out-of-the-box thinking that can push an entire industry forward. I'm very glad Kilobaser and myself can be part of this initiative. Of course, this presentation wouldn't be complete without a quick mention of what Kilobaser contributed to the field. The idea for Kilobaser's microfluidic chip is based on a series of microfluidic papers published by the Quake group around 26. Unfortunately, microfluidic technology is infamous for being hard to mass produce. If active actuation of liquids is embedded into the chip, things become even more difficult. And for DNA synthesis, the whole system must also be resistant against harsh solvents, or they need to be substituted. So what we did is we transformed the concept to injection molded polymer chips. The liquids are actually actively controlled with pneumatic on-chip membrane valves. The benefits of all this is that the fluidic channels are embedded in a disposable chip. Everyone who ever operated a classic synthesizer knows that the tubing and valves need constant attention and maintenance. Not with the chip. This is just thrown away after synthesis. And since the distance between the column and valves is much smaller, and there are no tubes to fill and empty with each cycle. It also needs a lot less reagents. The overall volume of reagents needed for 150 basis synthesis is only around 18 milliliters. So we are able to store all reagents and even the waste bottles inside a single compact cartridge. This means that scientists without experience in chemistry or even robots can operate the device. Another key innovation was the integration of cleavage and finality protection steps directly into the device. So a usable product comes directly out of the machine. This means also a robot, like the microfactory developed within our dynamic consortium, can access and use freshly made DNA, fully autonomous and without human intervention. This looks like this. And with that, I'd like to hand over to Mark, who will now take us from the single oligo to the library scale. Thank you very much for your attention.
Okay, I hope everybody can hear me. I'm going to share my screen.
Yes, we can hear you and see your slides.
Okay, great. So, okay, I'm going to thank you, Marvin. I'm going to take over and discuss the transition from solid phase synthesis to large scale. So I'm not going to talk about the chemistry in any detail. The chemistry remains largely unchanged. It's not 100% changed. And I'll discuss a few of the details. But the main thing I want to do is I want to sort of simplify the chemical cycle into two key components, as Marvin discussed. So there's a D block component in which we remove a protecting group from typically the 5' carbon of the sugar, or it's also possible to do it, reverse everything and do it from the 3' side. But let's stay with the 5' side. So if you look at the red structure here, that is the protecting group that is commonly used for solid phase synthesis. There are other options. I'll come back to that later. But we're going to sort of neglect the rest. So we have essentially simplified this to a coupling side and to a side which is temporarily chemically deprotected. And all strategies related to large-scale synthesis involve either addressing the D block side or the coupling side, but never both. So again, the idea is, as Marvin said, is we're going to just sort of do these chemical reactions, these cycle-based cycle-based phosphoramidite chemistry, and we're going to add nucleotides one by one until we get an oligonucleotide of the length we want with the sequence we want. And then the question is, how do we scale this up to synthesizing hundreds up to millions of different oligonucleotides in parallel? Okay, so there's another key difference between solid phase synthesis and large-scale synthesis. So Marvin showed you a couple of pictures of a column. So this is where the solid phase synthesis takes place. There's different kinds of columns. In this case, it's a so-called expedite column. And we flow the reagents through this tube, and then there's a filter on the top and a filter on the bottom, and these beads that Marvin showed you in the middle. And we do the chemistry in these and the sort of very porous beads that are in between the two. Large-scale synthesis is exclusively done on flat surfaces, typically glass, but it could also be plastic or silicon or metal or carbon, like amorphous carbon or diamond-like carbon. But the main thing is that they're flat, and so really relatively simple geometry, and also that we have to do some chemistry on the surface to make it reactive so that the coupling reaction works the first time. So after the first nucleotide is added to the surface, we have the 5' hydroxyl group of the nucleotide, but initially we have to add some reactive surface group to the surface. It could be a hydroxyl group, but it could be other things as well. I won't go into that, but since we start out with a glass slide or another material and do some chemistry to add some reactive groups to the surface, and then we can proceed. There's two strategies, and only two, I think, that I've ever come across for large-scale synthesis. And they both involve spatial control of some of one of the reactions. So we can either choose to control spatially the deblocking step, or we can choose to achieve spatial control of the coupling step. You don't need to do both, because once you do one, the other one sort of follows. And so there's lots of other cycles that are involved in synthesis chemistry, but those additional cycles, so in the case of spatial control, deblocking, everything else is just done by flooding the surface. And in the case of spatial control of coupling, all the other reactions other than coupling are done just by flooding the surface with the appropriate solvents and reagents. Let me talk first, do this in sort of reverse order, and first talk about strategies for spatial control of coupling. And I start with this just because there's only one approach which is in use. And so it's less common, but that doesn't mean it's worse or inferior in any way. In fact, it typically leads to the highest purity of oligonucleotides. And this is practiced commercially by Agilent Technologies. So they originated, and then it was sort of taped. Then Twist was a sort of like it was on a spinoff, but people from Agilent then formed Twist, and they now sell large-scale oligo pools. But let me tell you about how this works. So again, this is spatial control of coupling, and this is the only example of spatial control of coupling. And the idea is here, you take a glass surface, and you give it some sort of chemical functionalization. In the case of this type of synthesis, it's a relatively complex surface functionalization because you need to pattern the surface such that you have, well, the whole surface is essentially hydrophobic, and then you have little circles that are hydrophilic. And the reason for that is because you're going to squirt little droplets of reagent onto the surface, and you want them to sort of form these nice little round circles. And you can sort of enforce that by making these sort of patches on the surface that are hydrophilic, because DNA is hydrophilic, and the phosphoramidite that we're using for the reaction is also hydrophilic. So essentially, you use well, both Agilent and Twist use commercial, I think they're made by Canon. They're inkjet printer engines that you take the existing inks. I'm sure other modifications are necessary as well, but you replace the 4 inks plus black with ACGT phosphoramidites, and then you just print on the surface. So you do one layer, and then you do some chemistry on the whole surface, and then you sort of one by one squirt out the little phosphoramidites on the appropriate dot, and then you do some more chemistry, and you repeat this potentially hundreds of times, maybe up to 400 times. Twist probably has to, to make a 200-mer, you would have to do this 200 times, for example. And then at the end, you do a little bit more chemistry, and then you either leave it as a microarray. So for example, Agilent primarily sells microarray like gene expression microarrays and others. Or in the case of Twist, you can cleave the DNA from the surface and then sell it to the customer as a custom library. So that's it for spatial control of coupling. So in the case of Agilent, we're squirting the phosphoramidite, which does the coupling reaction. And the other option is spatial control of deblocking. So I mentioned that on the 5' carbon of the sugar, there's a protecting group. In the case of solid phase synthesis, it's a acid labile protecting group. So if you expose it to acid, it falls off, and then you have the 5' hydroxyl, which is reactive to the incoming phosphoramidite. So there's 3 general approaches to achieve spatial control of deblocking. One is photolithographic, in which we replace this acid labile protecting group with the photolabile protecting group. Another one is electrochemical, where we use electrochemistry to generate an acidic environment that then removes this acid labile protecting group. And the other one is something like a hybrid between these two. So we use light to create in a specific type of reagent solution a photoacid. So we essentially use light to catalyze the conversion of a molecule from a neutral molecule to an acidic molecule. And then that acidic molecule removes the 5' blocking group on standard phosphoramidites. And then we can continue with the spatial control of the reaction. So let me do these one by one. So the first one, photolithographic synthesis, was pioneered by Affymetrix. You can see it was roughly 1992, which is about 10 years after Caruthers developed the phosphoramidite chemistry. So it took about 10 years to go from solid phase synthesis, or essentially modern solid phase synthesis, to, well, essentially modern large scale synthesis. Originally, Affymetrix tried to make peptide arrays, but that turned out to be more difficult than they bargained for, and so they pivoted to making DNA microarrays, which is chemistry is significantly simpler, and also because you only have four monomers, it's easier than peptide synthesis. You can see here from the picture that it's a relatively complex sort of industrial environment necessary for the production. They use 1980s era semiconductor photolithography equipment, and so they synthesize on wafers using steppers that were purchased from that were originally intended for microchip synthesis. But the key change to the chemistry was to introduce this MENPOC group. Let me see if I can use the laser pointer. So up here, this MENPOC is a photolabile protected group, and it replaces this DMT. So you have the four DNA monomers, each with a MENPOC group, and then you use a light source and you shine it through a piece of metal that has holes etched out of it, and they reach the surface, which is sort of a wafer equivalent surface, and you selectively remove the photolabile group from various parts of the surface, and the whole scheme is shown schematically over here. So you have an initial surface that has the photolabile groups. You selectively remove some of those photolabile groups, and then you have the reactive group, which is a hydroxyl group, and then you add the incoming phosphoramidite, and then that couples to the hydroxyl group. And you repeat this hundreds of times. Well, in the case of Affymetrix, I think they limited themselves due to relatively poor characteristics of the MENPOC group to roughly arrays of 35 mers. So that requires on the order of 100 cycles of coupling reactions. Some years after that, not like roughly 10 years after that, there was an innovation at the University of Wisconsin. So Franco Serena, he was a physicist, and he got together with some biologists and some chemists, and they came up with an analogous system, but instead of using complex industrial machinery, what they did is they built an optical system also with a light source, but instead of using physical masks that you shine light through, they introduced the use of these digital micromirror devices. So these are devices that have arrays of micromirrors, and they're tiltable in electrically tiltable. So you can do this essentially in software. And other than that, the process was the same as that used by Affymetrix. But the other thing that they were able to do, by then, a new photolabile group had been invented. This was at the University of Constance in Germany, this NPPOC. And the NPPOC group just works much better than MENPOC. And so the synthesis equipment was much simpler because you could make a tabletop device that did this. And also the chemistry was much better, especially the photochemistry. So you could achieve like 60 mers instead of Affymetrix could only get like 30, 35 mers. With a new approach, you could make 60, 70, 80 mers, maybe 100 mers. And so there was a spin-off from the University of Wisconsin called Nimblegen, and they commercialized this type of approach to making microarrays. And at the time, this was before synthesis, before people were using libraries typically. So mainly the main application was gene expression arrays and other types of large-scale genomic assays, but they were based on microarrays. So essentially uncleaved surfaces, DNA on surfaces, but uncleaved. So Nimblegen was founded. It was eventually sold to Roche, and then Roche closed it in 2012. During that period, also they were able to improve the photolabile group from the NPPOC group to the thiophenyl NPPOC group, which is the sort of current state of the art. The main difference between them is that it takes about 10 times less light to remove the thiophenyl and PPOC. So this deblocking reaction with thiophenyl and PPOC can be done in like 3 to 6 seconds rather than a minute. Roughly at the same time, there was a company in Germany that also sort of got on the bandwagon of mass photolithography. It was called Fibit. They didn't make it for very long. And there's not a whole lot of information available anymore about them. But I was able to find a couple of pictures. So one of the surface. So again, it's a small piece of glass and some sort of a chamber. And the machine is a sort of enormous, I guess technically still desktop machine. But this was a multi-purpose machine. So the machine would not only do the synthesis of the microarray, but it also do the hybridization and readout of the hybridization data. So it's a complex, it's actually also made in Vienna. This machine is made in a company in Vienna. And so you would feed in this chip. It would have a pumping system inside, do the chemistry. It had a digital microarray device inside. It would do the full chemistry, it would do the protection. And then, I don't know the details, maybe you have to take it out and then do hybridization outside, or you just add the hybridization solution and do it inside. But it would do the hybridization, so it had some sort of oven inside. And then it would also read out the hybridization data within the machine. But anyway, so this is what was an alternative approach to that of Imogen, but with a much more complex machine that did other stuff as well. A few years after that, and in this general same direction, there was a company founded in Leiden in the Netherlands called FlexGen. Also, the chief scientific officer had come from the University of Constance, so this was an environment in which they were working a lot on photolabile groups and DNA synthesis. So what they came up with was an approach to use a UV laser, like a 365 UV laser, to do the photodeprotection. So the process is exactly the same. Instead of using a mask, physical mask that you shine light through, or using a digital micromirror device, you raster a laser over the surface. You switch it on and off, so that you deprotect at specific locations on the surface, and you alternate that with coupling chemistry. And then after whatever 100, 200 cycles, you have an array of different DNA sequences on the surface, which you can then cleave if your process calls for that. And they also survived for a few years, but I don't think they ever had a viable product. They struggled with this. And so for a long time, I thought maybe it was not a good approach, because maybe there's something about using this really high intensity lasers for a short amount of time instead of using a short illumination for a long period of time that was somehow detrimental to the DNA. But anyway, last year we were able to sort of make a similar device in our lab, and that works. So this is a rendering of that. You can see the advantage is very small. It's about the size of a shoebox. And again, has this UV laser. In this case, it's a 405 nanometer laser. We shape the beam, and then we can sort of raster across the surface, and there's a lens. And we turn it off and on, and you can see the outcome of such a synthesis, which is on the order of a few thousand oligonucleotides on the surface. And this is another example of using photolabile groups to create complexity on surfaces. Another alternative approach, this is sort of this hybrid approach where you use both chemistry and photochemistry to achieve deprotection. And this was pioneered commercially by Arbor Biosciences in Michigan. And it was based, I think, on research done at the Michigan one of the universities of Michigan. I can't remember the name. And the idea here is that this diagram is not very clear, but basically you have a glass surface. Inside some sort of chamber, you fill that chamber with a photoacid. And this is a solution that has a chemical in it that absorbs light, and when it absorbs light, it creates a hydrogen ion. So you can see those here. And then the advantage of this is that then you can create a local acidic environment. And then you can use regular DNA phosphoramides with the acid labile protecting group instead of the phosphoramides with the photolabile protecting group. So it's sort of a hybrid in the sense that you can use regular DNA phosphoramides, which are cheaper and more widely available, but you still achieve the spatial control by essentially a sort of type of photo deprotection, which is somewhat indirect because first you have the light, then you generate the acid, and then the acid does the deblocking. So this approach is, I mean, commercially viable, and I think you can buy libraries from them, for example. But I think the main disadvantage of this general approach of using hydrogen nuclei essentially to do the deblocking step is that H plus is a subatomic particle. It's a proton, and it diffuses extremely fast in any medium. And therefore, it's hard to sort of contain. So it tends to sort of travel far very quickly. And so you're sort of limited in the size of the DNA feature that you're trying to synthesize. And this is, I think this is somewhat more complex than the others because you have sort of a combination of two approaches. But nevertheless, it's an attractive approach because it allows you to use the inexpensive chemistry, the standard phosphoramide chemistry, but still have this very relatively good spatial control using relatively simple optical systems. So this leads me to electrochemical deblocking. So this is sort of similar in the sense that we're going to use hydrogen ions, so positively charged particles, to do the deblocking step. But in this case, we don't generate with photoacid. We generate them by having an anode and cathode in close proximity, so generate essentially an electric field at a small localized environment. And so this was sort of pioneered commercially by Custom Array, and Custom Array was then bought out by GeneScript a few years ago. And again, the chemistry is shown here, the electrochemistry is shown here. So you have anodes and cathodes, and then you use this, you add this molecule, hydroxyquinone, and then in the interface between this anode and this cathode, you have this electrochemistry. That electrochemistry generates a positive hydrogen ion, which then does the deblocking step, and then you have essentially regular phosphoramidite chemistry. So this approach works well, and they've been successful in making our microarrays and libraries. I think, Robert, you've used some of their libraries as well. I think the downside of this approach is that the chemistry is simple, but the substrate is very complex. So you can see here on the left, you need essentially a large-scale semiconductor substrate that has been made in a large fab that has hundreds of thousands or maybe millions of different individually addressable anode cathode pairs. So that you have to have one transistor, at least one transistor for each of these. And so the surface itself is very expensive and has to be made in very specialized facilities. And I don't know to what extent they're reusable, but the initial, as opposed to most of the other methods, the complexity and the cost of the synthesis is in the substrate itself rather than the chemistry. Okay, then let me get to the sort of final approach today, which is actually what I do. So what I do is essentially the same as what Nimblegen did. So maskless photolithography. Let me just go into that in a little bit more detail because that's what the approach we're using in the context of the DigNA project. So the original approach used by Nimblegen and then Jost Nimblegen was to use an optical system that's shown here, and this is just a curved mirror or two curved mirrors, and we have a digital micro mirror device at one place, and we illuminate it, and then the reflection from the mirrors that are tilted hit one optical surface, reflect to another optical surface in the back, and then they're imaged onto a glass surface. And that glass surface is where we do the spatially sensitive deblocking with photolabile groups. And so a few years ago, we thought we'd try to do better than Nimblegen did and be able to use larger arrays of micromirrors. So we wanted to use the 1080p or possibly the 4K DMD. So the original Nimble-Gen microarrays were made with half XGA, so roughly, I think, 400,000 mirrors. And then they switched to the XGA, and then I think at some point, towards the end, they switched to 1080p. But we wanted the option to go to even higher mirror densities. So these are not commercially available yet, at least not for our purpose. But in principle, one could use a 4K array, which has 8 million micromirrors, and then do really massive scale synthesis, which would be very useful for digital storage in DNA. Anyway, for this purpose, we developed a new optical system, which is shown here. It's a cathode optic relay, which we have the DMD, a lens, another lens, a reflective surface, and then we form an image over here. And the advantage of this system relative to the previous one is that we can accommodate much larger, much higher resolution synthesis. So we go from roughly 800,000 independent synthesis to on the order of 8 million, although we're not there yet. So routinely, we only do 2 million parallel synthesis of 2 million oligonucleotides in parallel. And so this is actually just the sort of a scheme of the system. Here on the bottom is a rendering of what the actual optical system looks like. I can show you a little bit more detail. So originally, the photodetection was done with a mercury lamp. Now we have high-powered LEDs that we can use. So we gather the light from the UV LED. We image it onto a homogenizer. This is just a glass tube. And then the light sort of refracts off the surfaces, and then it sort of spatially mixes. So you get like a rectangular beam of light coming out. And then we collect that, and then we image it onto this digital micromirror device. In this case, ours has 2.1 million mirrors. And the mirrors have two positions. One is an on position, one is an off position. The mirrors in the off position direct light to this sort of black surface where they get thrown up, the photon gets absorbed, turned into heat. The light from the on mirror bounces on this mirror, goes to this first lens, the second lens reflects on this mirror. It's a spherical mirror, and then back to the second lens, and then to the first lens, and then is imaged onto a flow cell where we do the chemistry. So this is an actual rendering of the system. So you can see here on the bottom left the picture of it. So it's not quite a tabletop device. On the top is the optical system. Below it is the fluidic system. So this is sort of like a very old version of the Kilobaser. This is one of the first DNA synthesizers that was sort of made at a large scale. They made thousands of them. It's called an Expedite 8909. It dates from around 1990. So this is now, it's like 35 years old. But it's a sort of reliable device that's been sort of the workhorse of the DNA synthesis community for many, many decades. The actual device is shown here. This is also a rendering. So since we have the LED over here, then we collect the light, and this is the homogenizer. Then the light reflects here, and the DMD is not visible, it's over here, but here you can see the control circuit for the DMD. And then the light reflects to this mirror here, goes through this first lens, the second lens, a mirror here, back, and then the flow cell is in the back over here. I just want to briefly talk in the last couple of slides about applications of this kind of photolithography. We call it maskless photolithography because as opposed to the original Affymetrix system, we don't use physical masks. So a mask, the kind that's used in photolithography is like typically a chrome film on glass, and the chrome is selectively etched away so that you have like windows that allow the light to be transmitted. But the problem with that is that every sort of mask has to be different. So you have to physically have this piece of glass with chrome on it, and then put it, store it, put it back. You have to make them, and you may need like several hundred of them for a synthesis. So maskless photolithography only means that we're replacing the sort of large collections of custom-made masks with a single digitally controlled array of micromirrors. So the advantage is that it's versatile. Everything is done sort of in software. There's essentially no moving parts except for the fluidic system. In the context of data storage and DNA, it's sort of a nice option because we can have lots of we can choose different ways of making DNA. So in the context of the DigNA project, we're making like composite DNA, we're using primers. We can also use like extended alphabet. So we can use, for example, non-ACGT, like so invented bases that people have come up with over the years that may also base pair with other artificial bases. And so in principle, you can also extend the sort of genetic library beyond the sort of the 4 canonical bases. But in our lab, we also do a lot of molecular biology research. And so this is a useful device to have for that. So we do a lot of transcriptomics, a lot of spatial transcriptomics, RNA binding proteins. We study, like we can make RNA. So again, for RNA synthesis, the chemistry is essentially identical. It's also phosphoramidite chemistry. The only difference, well, the main difference is that RNA has the 2' hydroxyl group, which also needs a separate protecting group. But other than that, the synthesis chemistry is the same. So we make RNA. We also do a lot of synthesis with non-canonical, both natural and artificial nucleotides. And so we also do, look at transcription factor binding and also CRISPR-Cas. I'll talk briefly about that in the next slide. But here are some examples of nucleotides that are sort of non-canonical nucleotides. This is 5-hydroxymethylcytosine, which is present in the DNA, especially in neurons. And so we can use that. This is L-DNA, which is a engineered nucleic acid, engineered nucleotide that has this bridge on the sugar. And this bridge makes it RNA-like, so it enforces RNA-like sugar conformation. And okay, I won't go into the details. And this is, for example, an abasic. So it's a nucleotide that is missing the base. And these are present also as an error in the genome. And so in the case of, for example, looking at transcription factors, you want to know how do these abasic sites affect transcription factor binding. I alluded to this at the beginning. We can also do synthesis backwards. So traditionally, DNA synthesis perform 3' to 5'. But if you sort of reverse everything, you put the DMT group on the 3' side and then the phosphoramidite part on the 3' side, you can synthesize in the opposite direction. This is generally not done because it's more expensive and a little bit harder, and it takes a little bit more time. But in some cases, you need to do that, particularly in the case of spatial transcriptomics. But anyway, there's lots of other, and okay, this is RNA. So we can also do RNA synthesis. Here's another example of a artificial nucleotide. In this case, we put a fluorine on the 2' carbon, and that makes it RNA-like. But so it behaves a lot like RNA, but it's much more stable than RNA. So in some applications, that can be very useful. So finally, let me just show you one particular application that we're working on, and this is to show you some of the versatility that you can achieve with the large-scale synthesis, particularly if you have full control over the sort of chemistry. So we're looking at the chemical modification space of guide RNA in CRISPR-Cas gene editing. So as you probably all know, CRISPR-Cas is used, I mean, it's a bacterial immune system, but it has been sort of repurposed to edit human or other DNA. And but one of the characteristics of it is that it needs RNA to sort of find its target. And RNA is always tricky to use therapeutically because it's very fragile. And so the idea is that most RNA-based therapies, you want to replace as much of the RNA as possible with RNA analogs. So these would be sort of chemically modified nucleotides that behave like RNA and are sort of recognized by enzymes as RNA, but are more stable than RNA. So in this particular project, the idea is to make a microarray that has, well, starts out with DNA, then we switch to RNA and RNA analogs in the guide part. And so this guide part would be the same sequence, but it would have different types of chemical modification. So instead of, for example, RNA-A, we would try fRNA-A or lRNA-A. And so to preserve the sequence, but change the sugar of the nucleotide at each position to see where the CRISPR-Cas enzyme works better. where it achieves the highest specificity, and so forth. Anyway, so we make this sort of hybrid DNA, RNA, modified RNA nucleotide on the surface, so not just one, but maybe a million of them, different sequences at different positions. Then we add the target for the CRISPR-Cas. So this would be a pre-made using solid phase synthesis, and it would have a 5' sorrel on one side and a 5' Cy3 fluorescent dye on the other one. This blue part is partially complementary down here, so they hybridize. And then the sorrelin intercalates, we expose it to UV light, it cross-links, and then we create one molecule that essentially has 3 strands. 2 RNA and 1 sort of mixed RNA, DNA, modified RNA. And then we add the Cas enzyme, and then the Cas enzyme sort of grabs everything. And then depending on how good our choice of the chemical modifications that guide RNA, it works better or worse. And if it works really well, it cleaves with high efficiency the target DNA, and then we lose the fluorescence. And then we can sort of look at the loss of fluorescence as a function of the particular choice of chemical modifications that we used. And this is on the right here is sort of a selection of the types of modifications that we'd use. So maybe the guide RNA tolerates DNA, prefers RNA, but we can do some selective substitution with DNA, 2' fRNA, 2' O-methyl RNA, LNA, and some other chemical modifications. Anyway, that brings us to an end to the presentation, and I hope it was helpful and gave you some insights into the chemical synthesis of DNA and also other nucleic acids and also some other applications.
Great, thank you very much, Mark and Martin, for the very nice presentation. There are a few questions, some in written, but first we'll try Tomasz Karcznik has raised his hand. I think Miriam can allow you to speak. Yes, please, Tomasz, go ahead with your question. You have to turn on your microphone. Tomasz, we don't hear you in case you are asking your question. Hello? Okay, no problem at all. Then let's go in the series of the presentations. There was one question from Mark in the chat by Yakini. He wanted to know a little more details about the Kilobaser specifications that may be of interest to the people here. What lengths do you have? What flexibility does the system offer?
Yeah, of course. Thank you, Sarah, for the question. If you don't mind, I'll share my screen once again. So
as you do, I'll just note that you can also ask questions by raising your hands, and then we'll allow you to talk if that's easier for you, or you can type them in the Q&A box, whatever is more convenient for you. Yeah.
Yeah. Exactly.
Yes, Martin, sorry for interrupting.
No, so the quick overview of Kilobaser properties are it's a single column device. So you have 1 oligo per run, and 1 run takes roughly 1.5 hours to achieve the final product. So after 1.5 hours, you get a 20-mer primer, or after 2 hours, you could get a 20-mer probe, TechMan probe, labeled on both ends. Length is recommended only to go up to 50 bases, because after this, you would need to employ some purification techniques, which are not embedded into the device. But up to 50 bases, especially for PCR applications, we say that the product can be used right out of the device for any downstream applications. The yield is roughly 300 picomores, which is sufficient for a couple of PCR reactions. The single column thing is what we are currently working on with Mark II in the course of a booster grant. So we are trying to also explore combining our techniques and integrate photolithography with our automated technology, actually. So in terms of flexibility, since we have this cartridge system, we can and do sell different combinations of reagents. So you can do different types of oligonucleotides. on the same machine, but you don't have to take care over the specific combination of chemistry you have to purchase. So on a traditional synthesizer, you would need to swap out certain chemicals if you want to do certain modifications. With our cartridge system, basically you can get the whole thing and everything needed in one single package. For example, fluorescent probes is, of course, the most commonly used modification, but we can also do some sorts of RNA, also like Mark mentioned, fluorinated, methoxylated RNAs, and few 5' phosphorylation and biotin modifications. So it's possible to get customized cartridges based on your requirements. We do it like that because we then usually would also modify the rest of the chemistry in order to match what specific properties your specific phosphoramidites, for example, would have. So that's a quick overview of the flexibility of Kilobaser, actually.
Then I see another question to the Kilobaser. If there's no purification, so yeah, that's one of the questions. How are the failed products removed?
Yeah. So purification is a very tough beast. We do provide some purification kits. Most others that are on the market are not specifically targeted at small oligos. So we developed our own to remove especially fluorescent dye. So in the case of probe synthesis, you would have some remnants of fluorescent dye, for example, in the final product. And this would disturb especially qPCR applications because you have an overwhelming amount of fluorescence in the product. So this is specifically developed to remove these fluorescent dyes. And we are also working on specific other purification kits for modifications. But yes, in general, our intent and goal is to be as straightforward as possible without requiring the customers to have expensive and very complicated purification equipment necessary on site.
Great, thanks very much. Then a few questions for Mark on, let's say, the array-based synthesis, especially the optical one. One question is if to what extent that the UV light exposure damages the DNA, or do you see any effects of that? And if you would add it would add a few minutes, but I'm curious how much damage, if at all, is caused by DNA in the UV exposure in the light-based deblocking step. It would add to a few minutes to exposure for long-mer synthesis. Yes, as what's the total UV exposure, and let's say what damage does it endure?
This is a complicated topic, and it's something that we're working on. At the sort of zero order level, there is none because DNA does not absorb at 360. I mean, so most of the absorbance of the DNA is at around 280 nanometers or under 300 at least, or maybe the tail of the absorption is under 320 nanometers. We do the exposure at 365. There is a little bit of anecdotal evidence that there could be the very long tail of the absorption of DNA, maybe reach 365. There should be none, but again, there's some anecdotal evidence that maybe there's some small amount of damage. We can also do the synthesis with this thiophenyl and PPOC, and then the synthesis, the exposure, light exposure is that it can be done at 405 nanometers, which is then sort of essentially visible light. And then there should be none because we're another 40 nanometers into the visible. Nevertheless, there are some issues, for example, with the photolabile group. When it comes off itself, remains able to absorb light. And one of the photolabile group, the benzyl and PPOC, when it comes off, it becomes what's a so-called Norris type 2 photooxidizer, or sorry, free radical photooxidizer, and then that may be sort of bumping into the DNA and causing some damage. So we're looking into that. We're doing we have some experiments underway to sort of try to elucidate if there's any sort of the second order type reactions that might be causing some damage to the DNA.
And then the other set of questions goes kind of in terms of quality. Let's say one is what's the current pitch between two separate spots that you can achieve with the current, let's say, optics you have?
So the two versions that we have at the moment is one is the XGA, and those are 14 micron pitch. There's a roughly a 1 micron gap between the spots, but that gap is smaller than the optical resolution of the system. So there's always some sort of leakage of light from one pixel to the next. So if you have two immediately adjacent pixels, there will be a fair amount of sort of crosstalk between them, and the error rate will be relatively high. So if we want a lower error synthesis, we can sort of separate them. We can sort of like, have each pixel of synthesis separated by the next pixel synthesis by sort of one unused mirror. And we can increase that as well. So we can have two unused mirrors. Optically, the diffraction doesn't extend beyond a few microns, but there's other optical effects of like scattering in the system from dust and stuff like so light can a small amount of light goes everywhere, and that potentially increases the insertion rate. And so there's always a trade-off between sort of density and the error rate. So if you want a low error synthesis, then you can't use all the mirrors. Maybe you can use a quarter of them or a tenth of them or something like that. In the 1080p, those are, I think, roughly 10 point something micron pitch, and there's roughly 2.1 million of them. We haven't done this so far, but there's also like the higher resolution DMDs up to 4K, and those have 7.6 micron pitches, and that would be I think 8.8 million mirrors. The error rate there would be fairly high. I mean, you would probably want large gaps just because also the mirrors are becoming relatively small relative to the resolution of the system. But anyway, and particularly for data storage in DNA where you can have error correcting codes, you still may be better off making the synthesis much cheaper, but having a higher error rate.
Yeah, because the other question goes in a very similar line. If you, let's say, scale to millions of oligos, what are the strategies of, let's say, reducing the errors that you have for enabling such high fidelity libraries? So would that be what you just said, or would you add other comments to that?
Well, so at the synthesis level, you can probably do some stuff to reduce the error rate, again, by reducing the density and things like that. I think one of the reasons, I mean, I don't know how it works, but I think like Twist and Agilent are probably library providers. They probably do some post-synthetic work up as well to sort of get rid of sort of truncations and other, and in principle, we could do that as well. We have not done that so far, but there are some sort of off-array post-synthetic steps that one can do to sort of clean up. One is like just PCR. If you do PCR cycles, you get rid of things that are maybe incomplete. But you could also use hybridization. There's some enzymes that recognize mismatches, and there's sort of enzymatic post-synthesis cleanup procedures that one could use to increase the purity further.
Thanks. One question that goes in a different direction is, would PNA, so peptide nucleic acid, offer simpler chemistry for synthesis?
Well, I've never done PNA, but I think PNA is harder than phosphoramidite chemistry is really probably one of the best chemical reactions that there is. It's very, the coupling efficiency is, up to 99.9%. It's hard. There are very few chemical reactions which are that good consistently. The forming peptide bonds is usually much harder. The reagents are harsher. The coupling times are minutes instead of seconds. So in general, PNA is, I would say, maybe a factor of 10 harder and less, so the yield would be substantially lower than for just phosphoramidite chemistry is just really great. And that's why, for example, Affymetrix, they sort of gave up on their peptide array synthesis and just focused on DNA array.
Okay, then I have a last question that goes to you both, very excited to see the photolithography and Kilobaser collaboration in the light of light-based deblocking straightforward on the solid support. Is the light-based deblocking straightforward on the solid support inside of Kilobaser's microfluidic chip, or would you need to make a custom platform for the NP-POC chemistry? So that's for either of you to decide how you want to answer that.
Yeah, Marvin, maybe you can handle it.
Okay, thank you. Yeah, so we are in the extremely early stages of exploring this. So yes, the final goal would be to integrate it directly into a single device on a single chip. Of course, it would need to look different than currently because you don't have a single small column, but instead a surface, which obviously changes the geometry of the whole system in some quite complicated ways. So yes, it would have to be a new platform to some degree, but it shares the same advantages actually that we can currently offer. So it would be a closed off, could be a closed off sealed system, which is can be mass produced and mass employed, also for large scale synthesis of for example, data storage. But we are also looking into ways to leverage on the work that Mark's team did with or the special modifications, special chemistry, because I think there's really a huge potential in this. While at the same time, it is exponentially difficult for non-experts to use. So we believe if we can offer a turnkey system which is able to use such advanced chemistry, this could be a very interesting product and could yield to very interesting applications within the medical field, especially.
Okay, thanks very much. I see no further questions. We're also well ahead of time. I still take the liberty and ask a question to both of you. Mark has shown so many examples of failed array synthesize companies. And still we buy oligos from service providers on array-based. Those seem to be more successful than, let's say, the companies trying to sell devices. Do you understand why that is the case? Or do you have an idea why that's so difficult?
Well, I think a few companies, FlexGen sold the device. Also, I think Customer sold the device. They don't do it anymore, but they sold it. I think most people don't want the complexity of doing the synthesis themselves, because most people who use DNA are molecular biologists, and they don't want to deal with the chemistry and there's fluidic systems that you have to maintain. There's a fair amount of complexity. So I think that's the sort of success of Kilobaser, is that they have a very simple fluidic system. But the fluidic system that we use, for example, I mean, we have to repair it essentially every week. And so it's sort of a hassle if you unless you really need DNA every day, twice a day, you're probably better off just buying it from somebody else.
Yeah, I actually agree. So I really also believe that it's only really feasible for end users to use technology, complex technology like this, if it's really off hands and it really just works. And this is definitely not the case for nucleic acid synthesis of any kind. And also, there are not too many companies left that make DNA synthesizers, like classic DNA synthesizers, because of the complexities involved in operating these devices, actually. So I really believe the concept we are chasing is the only way really to bring devices into the lab, even though it of course has advantages in speed and also versatility than you can just do with a local synthesis. But yeah, in the end, it's of course complex to operate if the manufacturers aren't really optimized for user friendliness. And yeah, in terms of,
let me just mention that we have made synthesizers, this massive array synthesizer for one group in Belgium and one for a company in California. And our concern was always like, how will they be able to sort of get it, to be able to use it effectively? And it involves probably at least one person who's sort of working full time on the device to sort of understand it well. And it's sufficiently complex that you can't just use it once a week or once a month. It has to be something that you use every day and have one person who really knows how to use it. Anyway, so we're also working on selling a couple more, but these are very specialized devices. You need to have the sort of a commitment to own and operate one, not just because of the cost, but just because you need some infrastructure. You need helium, argon. You need a room for them. You need supply cabinets and stuff like that. So it is a fairly large commitment to sort of do the synthesis yourself.
Okay, thanks. That gives a very nice end to our webinar. Also, I have to add that in all questions there was thank you for the very nice talk and the very specific one by Alexander Bittner, who said thank you for also thank you to both also for the historical perspective that was not just quite interesting, but also set a nice frame for those working more on the coding, storage, and analysis of DNA. So really to also give the history of this field was very helpful to many of us. And with that, I would like to thank you again very much for your presentations. Thank you everybody for joining. See you all, or most of you, soon again in Rome, probably at our next meeting. Yeah.
Thank you
Looking
very
forward
much.
to
Yeah,
that.
thanks
Bye-bye.
everyone. Goodbye.
Bye.
Bye.